This is the Sugar Science, and we're welcoming you to Ask the Expert, a brief, informative, and lively discussion with experts in the type 1 diabetes and related interdisciplinary research. We're recording this event, and we'll post it on the Sugar Science site YouTube channel shortly after presentation. If you have questions for our guests, please feel free to enter them in the chat or raise your hand at the end of the presentation. So today we have as our guest coming to us from UCSD, University of California, San Diego, Dr. Kyle Gaulton. And I'll give a little bit about him and then I'll let him talk about his sort of background. But he's an assistant professor at the Department of Pediatrics at UC San Diego. He has a BAS in computer science from University of Pennsylvania, PhD in genetics and molecular biology from University of North Carolina, Chapel Hill. And then he did postdoctoral training at the University of Oxford and Stanford University. So yeah, kind of hitting all the big universities and doing some really interesting work down there in San Diego. A lot of people are talking about this work. I would also just comment to say that I've been following the work, and during 2020, your lab was publishing like crazy. And that trend continued into 2021 with sort of like samples from the menu in 2021 were interpreting the type 1 diabetes risk using genetics and single cell epigenomics. That was in Nature. And then another one, systemic analysis of binding of transcription factors to non-coding variants, another Nature. And then single cell chromatin accessibility reveals pancreatic islet cell type and state specific regulatory programs of diabetes, and Nature Genetics. So these are really high profile journals, and that's really saying something, I think, about the work that you're doing. I think this particular paper kind of spells out the scale of what you and your lab have been up to, and then you can sort of dive in, after this. But I really like this paper. The large scale genetic association and single cell accessible chromatin mapping defines cell type specific mechanisms of T1D risk. It was in bioRxives in January 2021. And if I can just do a thumbnail sketch of it, the work was just massive. I mean, it was translating GWAS of a complex disease into mechanistic insight requires a comprehensive understanding of risk variant effects on disease relevant cell types. Yes, totally true. And you guys are wading into this fearlessly, I think. And so what you did was you combined sort of the genetic association mapping and single cell epigenomics. So that's sort of a finer line, right? And they performed the largest to date GWAS of type 1 diabetes in 489,679 samples, imputed into 59.2 million variants. From there, they kind of distilled it down. They identified 74 novel association signals, including several large effect rare variants. So then they did fine mapping and they got 141 total signals. And that really improved the resolution of causal credible variant sort of credible sets that they mapped to non-coding sequence. So this is so interesting. And I'd love for you to, sort of tell us what's going on in the lab, where you are, elaborate on that very cool paper, and yeah, just fill us in.
Sure. No, I'd be happy to. I was going to say that we probably published so many papers last year because we weren't doing much else, so we're sitting at home for.
Well, it's a good use of time.
But thank you so much. So I guess I can say very briefly a bit about myself. I mean, you touched on education, and I'll just say that. I was thinking this morning that I've been studying diabetes genetics for about 15 years, just turned 40. So pretty much ever since I started in grad school studying the genetics of diabetes, for a long time it was type 2 diabetes, both graduate school and postdoc. The labs I worked in primarily focused on type 2 diabetes, and when we moved here to San Diego, shift our focus a bit to type 1 diabetes. And I think the genetics of type 1 share some similarities to type 2, but many differences, and very interesting to have been able to try to use genetics to kind of try to unlock some of the biology underlying the trait. So I will share my screen now for my.
Yes.
Okay.
You should be able to.
All right, can you see my screen?
Yeah, looks perfect. Here we go.
So yeah, so thanks, Monica. I mean, you kind of detailed at a high level some of the things we're interested in our lab, and this paper that just came out in Nature recently, I think exemplifies a lot of our general interest for trying to combine genetic mapping of type 1 diabetes with mapping the epigenome of specific cell types that are relevant to the disease, and through the combination of those two, hope to learn something about the biology underlying the trait, genes involved in the trait, and try to give insight to maybe guide how the disease is potentially developing, and perhaps give some insight into potential therapeutics as well. So, okay. So I just have a handful of slides to detail this. So, so we study population genetics of type 1 diabetes. So to start, what we're talking about in terms of what we're studying in the top left are variation that exists between individuals. The primary sources of variation between people are called SNPs, single base pair changes. So that's the majority of risk of the disease, or single base pair changes between individuals, as well as what are called indels, insertion deletions, which are just short insertions or deletions of sequence. So that's most of what we study. It's not all the variation that exists in the genome, but that's primarily what we're interested in studying. And even though these create small changes, and in many cases just single changes in the genome, they can have profound effects on people's risk of developing disease. And the way we study these variants is it's actually a fairly simple paradigm, which is essentially to collect individuals who are cases, type 1 diabetics. People are controls. Most of the data we get are from public repositories. So all the data that was included in this Nature study was not genetic data we collected ourselves. It was either available in dbGaP or biobanks. And as the resources for human genetics have expanded, particularly for biobanks, the availability of these massive data sets enables this type of science. But so we have case samples, control samples, and then we're essentially comparing the relative frequency of the variants in cases versus controls. And then we do this for all variants across the genome, and that's what's being shown on the bottom, and that's what a GWAS study is.
Yeah.
So
can I just interject for 1 second?
Yeah.
So,
Yeah, sure.
So these, just sort of a shout out to sort of all the postdocs in the room. So these these repositories are available right now, and open for business basically. So people interested in type 1 can kind of approach them from different disciplines and different interests, and kind of utilize these repositories, and really kind of, I guess I want to say, go fishing, right?
I mean, these are really phenomenal resources. I mean, revolutionary. And the UK Biobank, FinGen is another massive resource. There's like Japan Biobank, there's other biobanks, and so these are massive collections of samples from population, where they have incredibly detailed phenotype information, genotype information in most cases. And yeah, I mean, and it allows you to study any trait you're interested in that surveyed in that population.
Right.
So, yeah, we're interested in type 1 diabetes, but for any trait you may be interested in,
Okay.
you can do that as well. And also they make a lot of the resources now, like you don't even have to do the analyses yourself in many cases. Like FinGen, for example, provides all of the summary data, so the results of the GWAS is for anybody to go get, so you don't actually have to redo all these studies from scratch. And they're also increasingly integrated in many other databases as well. So really, really incredible resources, and extremely valuable for understanding the genetics of these traits, and also, again, a lot of value for understanding therapeutic targets.
Yeah. I just wanted to kind of shout out to those resources. Thanks for elaborating.
Yeah, no, of course. Yeah, like I said, it enables all this work in ways that would not have been possible, 10 years ago.
Yeah.
Okay, and then but through these studies for type 1 diabetes, in this case, they've been fairly successful in terms of identifying risk loci for the disease. So as of last year, or a couple of years ago, there were about 60 loci known to affect the trait, which together explain about 80% of the heritability of the trait. Most of the heritability is explained by a single locus, MHC. And then there's a handful of other loci like insulin and PTPN22 that have reasonably large effects on disease risk. And then as you get beyond that, there's a long tail of variants that have relatively modest effects on the disease. So, and I say that because, so even though these 60 loci explain 80% of the heritability, probably getting up to 100%, like explaining all of the heritability of the trait, will encompass many, many, many additional risk loci that have probably increasingly small effects that are common. There's probably a lot of rare variants as well to define that have large effects, but rare variants don't explain much of the heritability because they aren't found in many individuals. So the general point is that there's a ton of genetic influence over type 1 diabetes.
Yeah.
And,
and it's quite heterogeneous, right, in the presentation. So it just
yeah.
makes it a little more complicated.
No, I mean, that's something that's of course being increasingly appreciated as well for type 1 and type 2 diabetes, that there's a lot of heterogeneity in the trait, and you know, thinking about genetic risk relative to different forms of the disease, different ages of onset. I mean, these are all really interesting questions that are mostly open. For sure, yeah. So I think as you start to separate the disease out into subtypes and things, that will further help clarify the genetic risk, probably identify additional loci for those subtypes as well.
Yeah.
But okay, but we're largely interested in exploiting the features of these genetic data, in that they're highly polygenic, so that many common, many rare variants contribute to disease. Outside of MHC, again, most of these variants have relatively small effects on disease, so there are rare variants that have large effects as well. The key thing is that most of these risk variants are non-coding, that you touched on, Monica, and likely affect gene regulation. And so this is, given that there's so many risk variants that affect the disease, and most of them are non-coding, that represents an incredible opportunity to take this information and overlay it with regulatory information for different cell types and tissues in the body, to try to find patterns, basically, for where these risk variants fall relative to where regulatory elements are. So we can exploit the extreme polygenicity of these traits and the fact that they're mostly non-coding, to use that to try to identify patterns that maybe tell us something about the cell types that are involved in the disease. So, and that's exactly what we did in this recent study. So in order to identify regulatory elements in the genome, I'll just briefly touch on one of the key techniques that we use to do that. So we're mapping the epigenome. So the epigenome is the kind of structure that sits around the genome that dictates when and where parts of the genome are being used, parts of the genome are accessible to proteins to regulate gene function. And one phenomenon in particular that we used in most of our work is called accessible chromatin. This is a hallmark of active regulatory elements, where the chromatin is basically exposed for regulatory proteins to come in and regulate gene function. And there's numerous assays that have been developed that can measure accessible chromatin as a readout for regulatory activity. The most popular one is ATAC-seq on the right, and that can tell you regions of the genome that are active. Now, up until very recently, that allowed you to maybe profile tissues or maybe cells that were sorted, like FACS sorting. But increasingly now, these assays are able to be done from individual cells, so the single cell versions of these ATAC-seq assays, which give you these epigenomic profiles from tens of thousands of cells. And so you can take the profiles from these cells, and one of the key analytical steps in working with these data is to try to take all these profiles and then derive the specific cell types that were within a tissue that you were assaying with these techniques. And then, you know, from there you can identify cell types, and then you can map the epigenome of individual cell types. And so that gives you regulatory programs in many cell types that you can then intersect with genetic data. And it's, again, the combination of the two that represent a really powerful approach to understand the cell types that are potentially involved in the disease. And this is from, on the right here, this is from a paper that's on bioRxiv that we were involved in, but mostly from our collaborators, Bing Ren and the UCSD Center for Epigenomics, where they did these single cell assays and I think like 25 different human tissues and mapped regulatory programs in many, many human cell types. So you can get extremely detailed information about the regulatory programs of individual cell types, which is very valuable in terms of then intersecting with genetic data.
Yeah. So if you were to, have access to a blood repository of newly diagnosed patients at age 13, for instance, you'd be able to isolate the APCs, or one APC and one T cell or whatever. Once you identified the T cell of interest, then you could run them through these series and be able to get a snapshot of what's happening at onset. Is that correct or no?
Yeah, exactly. Yeah. So you can take blood or any tissue, and these assays allow you to profile individual cells from blood or from the tissue, and then from that define the cell types that were there. So in peripheral blood, you could identify the immune cell populations that are present. In terms of identifying cell types and then subtypes, in terms of identifying perhaps like antigen specific T cells, things like that you can also do with single cell assays. Require slightly different assays. 10x, I know 10x is a sponsor, but 10x makes kits to profile, for example, like a T cell receptor in addition to, I think, RNA-seq, right? So you can even get information about like antigen specific T cells and stuff like that. But yeah, that's the general the general gist. Is that you can take tissue from non-diseased individuals, which is what we did in this study, to just get a general profile of like the regulatory landscape of cell types. But you can also use it to profile patient samples, and we have several studies doing that. Actually, mostly in pancreas, working with NPOD and Mark and guys at Florida, but also it could be done from peripheral blood.
Yeah, that's very interesting. We were talking with a group a couple of weeks ago about like that they would like to have more of a blood repository at diagnosis, so that these signatures could be captured. So yeah, this is really groundbreaking, and I think you're kind of like on a frontier here, setting this whole thing up. And I would also say that to the postdocs and the graduate students, this is like a really, really sort of cutting edge work and interesting projects here. And it seems like there's a lot of work to be done too.
Anyway, so to your question, Monica, so yeah, I mean, I think profiling blood banks and other tissue repositories using these techniques is a very, very promising way to gain access to information of changes that either occurred during disease or may precede disease in specific cell types. Mm-hmm. And the suite of assays for single cell genomics is really, really impressive. And I mean, the rate at which the field is changing and developing, and new types of assays are developed, and like paired assays, like information for different types of assays from the same cell. I mean, the development of these technologies has really been incredible, and they all represent an amazing opportunity to understand disease. Okay, so in our case, so what we we're doing is like I was saying, taking genetic information for type 1 diabetes and combining it with these cell type specific epigenome maps with the goal of integrating the two to try to identify cell types that may be involved in genetic risk of the disease. And so on the GWAS side, on the left, that study, like you mentioned, represents the largest genetic study of type 1 diabetes ever done. I think the previous GWASs before this were in the order of like maybe 10 or 20, 30,000 samples. So quite a bit larger in terms of number of samples, even though actually most of the samples are controls. So they're coming from like biobanks where, the population rate of type 1 diabetes is quite low. So most of the samples in a biobank are non-diabetic. But it's still quite a large number of samples that gives us greater resolution to map new loci, and in addition to specific variants of those loci that cause disease. Also, the breadth of variants that we're able to test now is increasing. Again, phenomenal resources that have been made available to predict genotypes in hundreds of millions of variants. There's a resource called TopMed, which is basically a service that you can upload your genotyping data to, and then they impute all these variants for you with the service, and you get the data back. So the resources for doing genetics are really incredible, both in terms of biobanks, but then also in terms of the ability to predict variants across the genome. So these 60 million variants, most of them are actually predicted genotypes, not actual measured genotypes, but they're in most cases very accurate.
Right.
So we combine the GWAS data with the epigenome map. So one question we then ask is do the variants that are associated with type 1 diabetes tend to fall in regulatory sites that are active in specific cell types from our maps? And so this is showing enrichment of certain cell types for risk variants for type 1 diabetes. So we see enrichment, as you would expect, for T cells, CD4 and CD8 T cells, other immune cells, PDCs or plasmacytoid dendritic cells, monocytes, beta cells, which are known to contribute intrinsically some risk of type 1 diabetes, and then also evidence for enrichment of cell types that perhaps haven't been implicated causally in the disease, as and ductal cells of the exocrine pancreas.
Yeah, and that's been, we had a great state of the science with some young scientists, Teresa Mastracci and Emily Sims and others talking about how the exocrine pancreas is really starting to show a role. And it's some fabulous work coming out of Indiana, and really, really great. So I'm very excited to see that they're here in your studies as well. The overlap is very interesting.
Yeah, I think there have been numerous studies recently that have kind of coalesced around the evidence for the exocrine pancreas being being changed in disease, but through these studies potentially having a role causally in the development of the disease as well. And of course, if you look back in literature and stuff, that there's long been observations of changes in the exocrine pancreas, right? So it's in some ways there, right? But I don't think had been either. I mean, either the technology wasn't there, or the ability to follow up on some of those observations.
Well, it is now. Thank you so much for doing all this excellent work.
Yeah. But yeah, I think this is one of many findings that are starting to emerge that suggests that the exocrine pancreas is playing a role in disease, in addition to the beta cells and of course immune cell types. And so that's a broad patterns, right? So we say broadly these cell types look like they're preferentially harboring risk of the disease, and so perhaps these are cell types that are involved in the disease. In the case of the exocrine cells, that was relatively new finding. And then we can also look at individual loci and say, well, what are the specific loci for disease that are driving those effects? Can we get a sense for, what genes may be implicated within these cell types to further, pick apart the mechanisms through which they may be contributing to disease? And in the case of exocrine cells, so we've highlighted different disease-rich cell types and then specific loci that kind of mapped in information for these cell types. And for the exocrine cells, there were numerous loci, many of which were fairly specific, like the risk variants were in sites that were specific to exocrine cells and often other cell types, which made us feel further confident that this is perhaps the relevant cell type here. And these loci were also linked to genes that in many cases have very exocrine specific function or expression, again suggesting that perhaps the genes are influencing disease through exocrine cells. So just some examples I've highlighted here. GP2 is a protein that comprises what are called the zymogen granules that are secreted from acinar cells that encode the digestive enzymes. It's also involved in, I think, innate immunity as well. CTRB1 and 2 are digestive enzymes. CFTR was one we were particularly interested in because it's quite famous.
Yeah, CFTR and the cystic fibrosis gene, right? So, I mean, that is really interesting.
Yeah, so CFTR causes cystic fibrosis, and then there's also a form of diabetes called CF related diabetes, cystic fibrosis related diabetes, which is increasingly common now that people with CF tend to live longer. And that's a form of diabetes that has thought to have been, so it's distinct from type 1 and type 2 diabetes, but kind of shares features in common with both. And so, yeah, so this suggests that perhaps CFTR is contributing to type 1 diabetes risk as well through ductal cells in that case. And at that locus in particular, we did a bunch of extra experimental work to show that like this site actually regulates CFTR in ductal cells, and validated that these predictive mechanisms a bit more. But on the whole, these loci and others that I'm not showing, suggest that the exocrine cells contributing to disease and highlight some genes and maybe some insight into through which these cells are contributing to disease. And so, but I think the question then is how does regulation of these genes and exocrine cells contribute to disease? We haven't proven that yet, right? I mean, these are patterns that we're observing, and it seems fairly compelling in terms of the genes that are nearby these variants. Like some of these genes are only expressed in like acinar cells, and so it seems fairly likely that those are the means through which they're contributing to disease, but I haven't proven in these cases. So the question is how regulation of these genes may contribute to disease risk. And like I said before, the long been reported abnormalities of the exocrine pancreas, and not only T1D, but autoantibody positive individuals, relatives of people who had type 1 diabetes, right? So these are changes that precede disease. In some cases, people who may never develop disease, but these are changes that are occurring before disease is happening. But I think A wasn't, quite known or appreciated, and maybe still an open question to what extent that's a consequence of disease processes caused by something else, or that's actually contributing somehow to the development of the disease.
Yeah, but it's so important to actually establish the etiology of of the disease in terms of the biology. I think it's 100 years after really the discovery of insulin, which is great, but it's also there's still so many unknowns about the actual etiology. So I think your work here is really pushing that, or opening several doors to really follow up on what is sort of like the timeline, what is the what are the changes, and how can we really appreciate how this thing manifests.
Right. Yeah. And so, yeah, no, exactly. Yeah. And thinking through the potential mechanisms through which they contribute to disease. So one thing we noted is that so many of the genes that are implicated are either digestive enzymes to CTRB1 and 2, and there's another gene, CEL, which is actually quite a famous gene in diabetes genetics, CEL, and which is another digestive enzyme, or they're involved in their secretion, CFTR or GP2 and things like this. And also we noticed that many of these same variants that are associated with type 1 diabetes risk also have some evidence for being associated with pancreatic disease. So like pancreatitis, pancreatic cancer, maybe like pancreatic cysts, but like, diseases of the exocrine pancreas.
One thing that's kind of weird is that I think that having type 1 diabetes is protective against having pancreatic cancer. That's like a weird nuance, but.
Yeah, right. I think for the, I'm not sure if the pancreatic cancer, I think for pancreatitis, I think most of the risk alleles for type 1 diabetes were also associated with increased risk of pancreatitis, but I think that was the case, but I actually don't quite remember. But yeah, the direction of effect, it was a bit off the top of my head, I'm not quite clear. I mean, this is the CFTR variant, so the directions are fairly consistent across these diseases, but for other ones there may be different directions of effect, but the variants tend to have some association in either direction with pancreatic diseases. Also in some cases with type 2 diabetes, which is another interesting question, the relationship between type 1 and type 2 that we're quite interested in.
Yeah.
But so to me and Natasha, it suggested that the potential mechanism through which these genes contributed to disease was maybe through promoting subclinical inflammation, like subclinical pancreatitis-like phenotypes, which could potentially promote immune infiltration, and then contribute to, in addition to many other risk factors, of course, the onset of disease. But that's speculation. But I think that that's now all to come in terms of understanding exactly how these genes are contributing to disease through the exocrine pancreas and why. And I think there's probably a lot of interesting biology to learn through this. So yeah, okay, so that's it. So the general gist is using large-scale genetic data, many risk variants for the disease, most are non-coding, leveraging the features of those data in combination with these really detailed cell type maps that together can give us insight into the cell types that are involved in disease. And as the maps get even more refined, larger data sets, I'm sure that the refinement of the cell types involved in disease and maybe even the subtypes of for immune cells in particular, their roles in disease will be further clarified. So just the last thing I wanted to highlight, and I mentioned this on the panel we had the other day, is that all this data is being collected in this large resource called the Type 1 Diabetes Knowledge Portal, which is both genetic data and also functional genomics data, so like epigenomic data, functional screens, like a lot of information that tells you about the function of the genome and also gene function that can be used to try to understand the function of risk variants associated with the disease and the genes involved. And so I hope people find this resource helpful.
Yeah, we've been shouting out about it. Is the ultimate goal to sort of create a consensome here?
Yeah, a consensome in terms of like, part of the goal is to just aggregate the data that exists, like in the genetic space for type 1 diabetes, for example.
That's great.
And also, genomic data. I mean, there's so much genomic data, epigenomic data, and there's so many regulatory maps in immune cells and stimulated conditions and other conditions and disease samples, non-disease, different types of epigenomic data. I mean, it's really overwhelming, actually, the amount of data that's available, all of which has value in terms of understanding genome function and then risk variant activity. So a lot of it is just collecting all this data but also making it available to people to be able to use in a way that's straightforward enough so that they can actually gain some insight from it, right? So it's collecting all this data, but then providing tools that make interpretation of all of it together more straightforward. And there's many other, I mean, this is like a network. It's not just type 1 diabetes, many other diseases under this common metabolic disease remit as well. So you can, learn more about, variants associated with type 1 diabetes. Are they associated with other traits and like knowing like the pleiotropy of these variants is quite useful as well.
There's overlap on several of the autoimmune, diseases and metabolic diseases. I wonder, so if for a new user, they can pretty much navigate this on their own. Is there any kind of guidance or help for them to navigate it? That's question one. And then the second question is, if they've created a repository of their own or some kind of data of their own, can they upload it and add to this developing portal?
So the first point, so yes, there are help pages and help tools, and then there's also in the bottom right here, there's a webinar that we had to release the portal, which details the functionality of the site, the different types of data that are going into it, both in genetic and genomic space. So that may be helpful for some people to watch it if they want more information about how to use the site. But yeah, there's also quite extensive help on the site itself to navigate. And there's there's numerous ways that you can interact with it. You could search for specific genes, specific variants, regions of the genome. You can look at the trait level. Like if you want to know, for example, for type 1 diabetes, like what are the cell types that have genomic information that are most enriched for the trait? Like there are disease-centric pages that let you look at, here are the most enriched cell types, maybe T cells and beta cells and maybe exocrine cells as well. Like so you can even get that level of information. So there's numerous facets of the site that allow you to access this information. I think the webinar would be a good way for people who are interested in learning more to get started, because that kind of details the high level features of the site and the data. If people want to add data, that is great. I mean, I think we're hoping to include as many data sets that are relevant to type 1 as possible in this
How
resource.
many do you have on there now for type 1?
So for type 1, it's our study that was in Nature, and then numerous studies from Steve. I mean, Steve's been involved with type 1 diabetes genetics for so long, and it's really one of the primary stewards of the type 1 diabetes genetics, and along with John Todd. And so they have numerous studies that they've put together over the years. They have a recent paper in Nature Genetics, which was multi-ethnic study. That data is there, and then they have like their previous studies from years before in there. And we're like, we actually go through literature and like find other genetic studies and like reach out to people to try to get their data as well. And but I'm sure we've missed some. So I think the genetic space that we have some data sets from mostly from our groups, but including more over time would be the goal. And also from biobanks as well, like FinGen and I think Japan Biobank released summary data for their phenotypes as well. So aggregating data from biobanks is also part of this goal. But if people have data, then please reach out to us. If they have genetic data as well as epigenomic data that they think would be useful from immune cells or pancreas or other relevant tissues, then we're really interested in we're working with people that have those types of data as well to include it.
Yeah, no, that's very important. This is a great project, and it's growing. And I think, with you and Steve Rich, and I guess from the Broad, Noel Burt and Jason Flanick, this is really, you're creating a really excellent toolkit.
Great.
Yeah. Oh, okay, good. So yeah, I just wanted to just sort of say, so just to change gears for one more second here, what do you make of the Fossman lab newest results presented at ADA? I mean, in terms of, it's a pretty big claim, BCG restores and resets the immune system and corrects the overmethylation of FOXP3. But just to speak to that idea rather than the data, what is your thought about that idea?
Yeah, so I think the idea of modifying the epigenome therapeutically is very interesting, and I think particularly as we find regions of the genome that are active in certain cell types, immune cells, beta cells, other disease-relevant cell types that you also know harbor risk variants for the disease. So you know that modifying that site is causally contributing to the disease. In most cases, those variants have quite small effects on disease, right? So that's always one of the criticisms of like human genetic work generally is that the effects are very small, but the variant itself may have a small effect, but that it's contributing causally to the development of the disease, which is useful information. B, you may be able through inhibiting the site or activating the site, depending on the direction the variant operates, to create a larger effect. Like maybe the variant has a subtle effect on the activity of the site and subtle effect on gene expression. But if you inactivate a whole site or you activate a site, that may create a more dramatic change that could have a much larger effect than the variant alone. But I think identifying parts of the genome that harbor risk that are potentially modifiable is a quite promising area for therapeutics.
Yeah.
And I think that those recent data, suggesting that methylation of FOXP3, is the mechanism through which that therapeutic is working. I don't know specifically too much about that, but I think that general the general area is I think very promising moving forward, particularly again for parts of the genome that are harboring risk, so that they have some causal role in the disease. To me, this seems like that they might make very attractive therapeutic targets.
Yeah. When I think of the genetic work, or I guess the genetic insults or fragility that's there, it almost seems like a lock on a safe. You dial and then you hit the number, and then you have to dial back and hit that number, and then you have to dial back and hit that number, and then the disease begins. So it's like a temporal plus a direct hit.
Yeah, and you need a lot of them as well. I mean, there's a lot of risk. Like everybody has some large combination of all these risk factors. I mean, of course,
Yeah.
there's, MHC, right? I mean, that's a different thing, right? But beyond the MHC, like all these additional risk factors, everybody's inheriting some combination of them, and yeah, it's modulating your risk in some direction. And there have been some really nice studies for other diseases that have shown, like on the extremes, like some people, by chance inherit a lot of risk alleles, or some people by chance inherit a lot of protective alleles. So those people can either be at very high risk for disease or very protective for disease, but that's a small percentage of total population. So yeah, for most people, it's again beyond MHC. I think it's dialing the risk in subtle directions. At the same time, I mean, something that may have people may be working on for type 1. I haven't really seen it too much, but it's possible that people working on this is trying to, like, say, for example, you find all these risk variants in exocrine cells. You think about, or maybe have, there's a lot of risk in beta cells as well. So maybe you take like individuals who happen to carry a lot of risk in beta cells, they may have a particular form of the disease because maybe they happen to inherit a lot of beta cell risk alleles, or maybe some people inherit a lot of exocrine risk alleles. And so that may be a way to kind of subdivide people based on, like,
put them into different baskets and then examine their trajectory, I guess.
Yeah, so that, I mean, that's worked a bit for type 2 diabetes. Again, I haven't seen it too much for type 1 yet, but you would imagine that, like, variants that affect genes within the same cell type, maybe there's some synergistic effects between all those genes acting within the same cell type, and that may put you in a certain risk category, and lead to different effects on complications. And even if it doesn't, even if it doesn't help you prevent the disease, it may be useful for understanding people's risk of complications later on, and how you might want to manage the treatment of their disease.
All roads lead to personalized medicine. I hate to say it because it's going to be tough to fund it. But anyway, so Tim Arthur is asking, Kyle, great work. I saw you have beta cells subsetted into insulin high and insulin low, and that there's the T1D GWAS enrichment in high and not in low. Do these beta cell subsets differ beyond insulin production? Do you think they could be distinguished in bulk RNA by cellular deconvolution? Same question for the other rare cell types that you pick up with single cell sequencing. That's a good question.
That's a very good question. So our other paper, this is in Nature Genetics, so that was largely the focus of that paper, was identifying these subsets of endocrine cells based on their epigenome. So for beta cells, there were distinct subsets that we found for they had many things that distinguished them. One of the most notable features that distinguished them was that for beta cells, like, one subset had high insulin activity, the other had low insulin activity. We also found the same phenomenon in alpha cells and delta cells, like low glucagon, high low somatostatin. But there were numerous differences between these subtypes, and it seemed like it was potentially related to insulin production or hormone regulation. Hormone production, hormone regulation, and then on the other side, maybe like signaling response or stress response. And so maybe thought this related to, like, subsets of cells they're perhaps making insulin versus those that were responding to some signal or undergoing some stress response. But that was all at the epigenomic level. So even though we tried to tie it to heterogeneity that's been described in gene expression, there was similar findings in single cell gene expression studies of different subsets of beta cells and other endocrine cell types. Newer studies where we have actually matched data between the epigenome and gene expression, I think will allow us to maybe resolve exactly what these different subsets mean at a resolution that we haven't quite been able to yet. But I think crudely, it seems like they're related to, like, hormone production versus signaling response, stress response. But I think understanding exactly what they're doing and how meaningful the heterogeneity is in terms of function are things that we're actively working on and interested in. For the other part of the question, so using that to deconvolute, bulk data, yeah, I mean, I think these profiles are very useful for deconvoluting bulk data sets, distinct subtypes of cell types, rare cell types. I mean, I think we've had some success in using these profiles to deconvolute bulk epigenomic data, and in a similar way, could easily be used to deconvolute bulk RNA-seq data. And I don't think we've tried it with these substates yet, but in general, it has been relatively straightforward for us to deconvolute bulk profiles using single cell maps.
Okay, thank you for that. And I know in the interest of time, I don't want to steal any more time for you, but there's one more question. Jennifer Wynn, she says, great talk. It's really nice to see evidence implicating exocrine pancreas with diabetes. Have you also seen whether T1D associated variants that are enriched in exocrine are also associated with T2D? That's cool too.
Well, that's a good question. So, one locus, CTRB1, so that is also known type 2 diabetes locus, and the effects of the variants are opposite. So the risk allele for type 1 diabetes is protective for type 2 diabetes. And actually for CFTR as well, I don't know if it's a known type 2 diabetes locus, but it has some significance for type 2 diabetes. And I think the effects, as far as I can remember, are opposite there as well. So some of these loci do affect both type 1 and type 2 diabetes, and at least seems superficially, just like looking at them so far, that they may have opposite effects on the two forms of the disease. There was a study last year, maybe the year before, that argued for a role for exocrine pancreas in type 2 diabetes. I can't remember the reference off the top of my head. But the mechanism there, I think, was through like exocrine signaling of beta cells. So maybe that like the exocrine pancreas is contributing to both forms of the disease, perhaps through different mechanisms, and so maybe that's why like the risk alleles may have opposite effects. But I think that it's certainly the case that there's some relationship there, and understanding that I think will be of high interest, and it seems quite complex given that in some cases appear to have opposite effects on the two diseases.
Yeah, maybe it's contextual. So yeah, I think this has just been amazing. Thank you so much for joining us and talking to us, and we wish you all the best in just moving the ball forward even further, which I think you're definitely poised to do, and in a great position to do. And I don't know, are you looking for postdocs? Maybe a shout out to our audience.
We're always looking for postdocs. I know Tim and Jennifer are grad students in the lab next door, so I know they're already
Fantastic.
a short hop over. But yeah, we're always looking for postdocs and people who are interested in genomics and genetics. So that'd be great. And like I said, for the knowledge portal, people who are interested in contributing data, who have genetic data sets, epigenomic data, functional data, like we're increasingly interested in like CRISPR screen type data, like that's very useful for understanding gene function and stuff like this. So anybody who wants to participate, I think that will only increase the value of the resource over time. So please.
Absolutely, it will. Yeah. Thank you again, and we'll hope to talk to you in the near future.
Yeah, thank you, Monica. Yeah, this is great.